So let's start the discussion on toxicology. And today, for 15, 20 minutes or so, we'll do a little bit of a historical perspective of toxicology and where it all stands. So again, this is the common slide across all presentations that I do, where we're stepping through the process from discovery to pharmacology to toxicology and to phase one. And as we looked at in pharmacology, pharmacology determines the right dose where you get optimal efficacy, studied through PK/PD. Toxicology will look at safety adverse events and determine what is called the maximum tolerated dose. So that's what the toxicologist will try to study. And then obviously the combination of the maximum tolerated dose and the efficacious dose is the dose window, which we are trying to determine. And the level of exposure, these terms are toxicology regulatory terms of the drug that you might have during the toxicology experiments.
Sorry to interrupt. Do you mind switching your video on just so that we get this
That's a good point.
kind of template every time? Right now we can see
it. Do that. Yeah. Okay. So the purpose of the toxicology study is to identify all the potential, not just the toxicities that you observe, but also potential toxicities that can be anticipated through these programs. We all know what adverse reactions are, and then you have severe adverse reactions. These are clinical terms that are defined in the regulations. So historically, in the FDA, the US FDA, the toxicology regulations started somewhere in the 1930s because of the potential use of this drug called sulfonamide. Sulfonamide was used for treatment of bacterial infections. And in the olden days, you would think you would go to a doctor and the doctor would give you some syrup to drink. It would be some combination of many chemicals. Compounders used to give these chemicals. And for any infection, you got the same, or any disease, you got the same drug, or same combination of drugs. But then at 1937, 100 people died. And that's when 1938, Congress enacted the Food and Drug Cosmetics Act, which required companies who are developing drugs to perform animal safety studies. Interestingly, the Drug and Cosmetics Act in India was established in 1945, which we still follow. So not quite far from, even before independence, but not far from what happened that time. Then about in 1957, the thalidomide story started unfolding, where children were being born to mothers who the children had no extremities. And this was primarily because of the side effect of, these are called teratogenetic effects, where this drug, thalidomide, was found to act, which was used as a painkiller during pregnancy, causes these deformities in bone formation during development, and these fetuses and the children didn't have the extremities. And that's when the next law came in, that you need to demonstrate that the reproductive safety, or the safety at the reproductive level. So reproductive tox came around in this phase. Thalidomide is actually quite an effective drug for pain. It has these teratogenic effects, but it has been approved later on for diseases like multiple myeloma and leprosy, where they're given to people who are beyond the age of childbearing age. So as long as not given in the childbearing age, it is actually still given. It's quite interesting drug that way. Then in 1982 was the Tylenol crisis, where somebody tried to tamper the packaging of the Tylenol and put cyanide in that. It was sort of almost like a terrorist, vicious attack. Knowingly bad things were done to Tylenol, and 7 people died. And that's where the regulations for tamper-resistant packaging became one of the regulations. This is very important in terms of that process. Then in 2004, erythropoietin, which is a growth factor for red blood cells, induced what is called PRCA, which is pure red cell aplasia, happened because patients getting erythropoietin, recombinant erythropoietin, started making antibodies to these erythropoietins and started getting autoimmune disease-like phenomenon. And a lot of investigation was done, and it was suggested that these stoppers, the rubber material in these stoppers, was going into the vial injection process and causing these aggregations, which could have been contributing to the immunogenicity of this recombinant protein, which induced antibodies. So then precise regulation for studying immunogenicity of drugs became a regulation. Similar. So the point I wanted to make with these 3 or 4 examples was that regulations don't come by just randomly. It's not like a bunch of people sitting in the room and saying, okay, let's put one regulation for this and that and the other. Usually it's a response, almost always actually, it's a response to something that has happened to the particular drug, and so they don't want something else to happen. And that's how these regulations have come about. And over the years, all of these regulations become cumulative, and everybody has to follow them. And so, as you can see, now everybody follows the FDA, all the regulation in the FDA. Like most countries try to follow what the FDA has done. The FDA has got more than 100 years of experience in terms of developing regulations, like I showed you here. And the other countries are catching up, so to speak. There's a lot of harmonization of regulations that are going on right now. And maybe that's a topic that we can talk about later at some point. How the regulations for toxicology in different countries work. But one nice thing is in the FDA and also other countries, but FDA for sure, is all the regulations, all the processes, all the standard operating procedure material, all of that is available on the FDA website. So if you want to understand how to submit a toxicology package, this guidance is very, very detailed. And it contains general toxicology, reproductive toxicology, and animal models. During the drug development process in an animal, in a toxicology study, in let's say a multinational company, a large company, big pharma, lots of steps are involved. All the toxicology department is doing a lot of work in different areas, right? They have a different group of people. The people who are working in the toxicology department have different skill sets. The ones that are involved in early toxicology development, where you're looking at omics technologies for finding out in vitro, in vivo, early small animal in vivo studies. You need molecular biologists, you need biochemists, you need assay development people at that time. Then as you move along the drug development path, you need people who are more, maybe veterinary science people, and people who know the regulations and who work in a more organized and say more regimented in a regimented place. And then that's how the talent for the various groups are recruited. Just a ballpark figure is usually about 300 to 500 preclinical studies can be going on at any given time in a in a large company. 100,000 rodents, 1,500 non-human primates, 1,500 dogs, 6,000 other animals, and usually the budget for a toxicology department in a large pharma maybe about $100 million, 50 to 80, $100 million. for all the studies that are going on. That sort of gives you an idea of the magnitude of the toxicology work that's going on. The S6 guidance, which is the ICH S6 guidance, is required, is a very detailed guidance for looking at toxicology of biological products. And so reviewing this one is is useful if you're going to study toxicology. Is there a chat? Yeah, gene signatures in vitro studies. Yes, Praveen. I'm going to pause in a minute after I finish this first part, and then we can have a little discussion. And as you can see in the table of content, you have preclinical safety testing, all of these things that I mentioned earlier, which is immunogenicity, dose selection. And then you also determine pharmacology, safety pharmacology, exposure, single dose studies, repeat dose studies, reproductive genotoxicity, carcinogenicity. All of these guidances are very clearly defined. As I mentioned earlier, the goal of the toxicity is to determine what is called that dose-limiting toxicity, determined by maximum tolerated dose, and you're determining what that window is. The points I've made here in this slide, which are platelet activation, pancreatitis, etc., are typical toxicology effects that you see for biologic or protein drugs. For small molecule drugs, the majority of them induce liver toxicity. And you don't see liver toxicity as often with recombinant proteins. So the kind of things, this is just to highlight the kind of toxicities that you see with recombinant proteins versus small molecule drugs. And as was mentioned in that guidance, you do all of these various toxicities all across the body when you do full toxicity. And again, from a job talent perspective, if you're going to work in a toxicology department in the company, you could be recruited to work in any of these subcategories of divisions. You could be looking at just cellular toxicity, for example, or immunogenicity, immunotoxicity. So kinds of skill sets that you need would be very different.